Okay, hi everybody. So we are just getting started in the parallel sessions number 2. So I'm very happy to be chairing this session. The first talk is by Mariana Marquez. Did I pronounce
Yeah,
correctly?
yes, that's fine.
Great. And the title of the presentation is Serotonin Enhances Responses to Envelope Stimuli via Burst Firing in Weakly Electrofish. You can hide the message that says Crowdcast is sharing your screen,
Oh yeah, yeah. Hi. And
and
then
then you can begin.
can you see my presentation?
Yeah, exactly.
Okay, okay,
Perfect.
okay.
Looks good.
All right. Yes. So I can start now. Okay. So in Dr. Chacron's lab, we study how neurons encode sensory input in order to generate behavioral responses. And as sensory inputs are very variable, it is important. Well, the brain uses neuromodulatory systems um, to adapt the neural responses according to the environment. So one of these neuromodulatory systems is the serotonergic system, which is highly conserved across vertebrates, and it's also present in multiple areas of the brain. So here, the big question for us is to understand how, if there is one main role of serotonin in sensory processing and what it is. So what we try to do is we use the electrosensory system of weakly electric fish. This way we try to answer this question. So weakly electric fish generate an electric field around its body, and that uses to navigate the world. So this electric field, it's called electric organ discharge, or EOD, and it's each fish can generate its own EOD. So if we were to put some dipoles in the water, we can measure the EOD amplitude as a function of time, and we see that this EOD amplitude varies as a sinusoid over time. And when two fish are together, we can see that there is an interference of the EOD signals that generate sinusoidal modulation of the electric field, which is here in number 1. Yes, there. And in a more realistic situation, when there's movement between the fish, this amplitude of this modulation gets further modulated and gives rise to what we call the envelope. So this stimulus, the envelope, has information about the distance between the fish. So the closer the fish are, we can see a peak in the envelope, and when the fish are further away, there is a trough. So what I did for this project was I recorded neurons that can encode the envelope, and these neurons are located in a hindbrain area. And with one electrode, I record the neural activity, and we can also inject serotonin with a pipette. So during baseline conditions without any stimulation, here I'm showing spike trace, and you can see that the neurons fire with isolated spikes or with burst activity. And what I saw after serotonin application was that there was an increase in the neuron activity and also an increase in the burst activity. And this was seen before in previous projects. So what I did later was to play the envelope stimulus and then record the neural activity again. So during control conditions, the neurons tend to fire at the peak of the envelope. So when you plot the fire rate of the neuron, you can see a modulation. But after serotonin application, it's it's enhanced. So I can quantify these results by measuring the gain, and the gain is the amplitude of the response divided by the amplitude of the stimulus. And when I vary the envelope frequency, I can build a tuning curve of the neurons. So during control condition, the gain increases as a power law with envelope frequency. And what I saw is that after serotonin application, the gain was increased for higher envelope frequencies. And so when I look further into the spike train of the neurons during the responses to the envelopes, so I split the train into the burst and the isolated spikes. And what I'm showing here is that the response that is calculated from the burst activity, it's modulated during control condition, and it's more prominent after serotonin application. But it's not the case for the isolated spikes. So I built the tuning curves from the bursts and from the isolated spikes, which is what I'm showing here. And as you see, the significant increases happened for the burst spikes and for higher envelope frequencies. And there's no significant change for the isolated spikes. So after doing this, I went and I recorded the behavioral responses. The behavioral responses can be measured with 2 electrodes in the water tube. And they consist of changes in the EOD frequency. So here you can see the envelope the stimulus that I played. And during control conditions, the fish EOD frequency tracks the envelope. And after bilateral serotonin application, this modulation is more prominent. So again, you can quantify this by measuring the gain as a function of envelope frequency. And as seen previously during control conditions, there is a decrease as a power law with envelope frequency. But after bilateral serotonin application, there is an increase for all envelope frequencies, that it's even more prominent for the higher envelope frequency. So this is consistent, and we saw a nice correlation between changes in the bursting activity and the gain calculated from the bursting activity and changes calculated from the behavioral gain. What's interesting is that when I did this, I compared these results with the different species of fish where I also did the experiments, and I saw that the neural responses were very similar, but the behavioral gain responses were different. So in that this Apternotus leptorhynchus species, the gain increased for lower envelope frequencies. So yes, so my conclusions here is that this is the summary of what I just told you. Serotonin increases cell excitability, enhances neural responses to envelope stimulus due to burst firing, and also enhances behavioral responses to envelope stimulus. And the behavioral changes are correlated with changes in burst and firing. And what's important is that the neural responses are similar between 2 species, but the behavioral responses are markedly different. So this suggests that the neural responses are decoded differently in downstream areas after serotonin application. So for my main question, whether there's a main role or the function of serotonin is species dependent, it seems from my results that it's species dependent. And that's it. So,
Great, thank you so much.
Yeah.
So we have time for questions. So
Stop. No.
let's, yeah, I can take away the screen if you like. Okay. So the first question is, what are the behavioral consequences of the serotonergic applications?
Yeah, so I don't know if I should go back to my slides. So this is what we observe when I immobilize a fish. I can record the changes in the EOD frequency. So when the fish is constantly changing a little bit the EOD frequency, their EOD frequency. So we don't know exactly why the fish is doing this, but we have seen that when you play the envelope, the fish tracks that stimulus, and we can only speculate that it has to do with how the fish wants the other fish to observe its own electric field. So yeah, we have observed, but we don't know exactly why.
Okay, great. And the second question is, what are the cellular effects of serotonin in the fish forebrain?
Yes, so how the fact that serotonin increases the neuron excitability, a previous work from my lab was done to study this, and they observed that serotonin there's like the after depolarization from a spike. Okay, so what serotonin does is that it decreases it inhibits sodium channels and causes the after hyperpolarization to decrease. And that's how like it facilitates the bursting activity.
Okay, great. Thank you very much. I'm going to invite the next speaker to the front. So here we go. Emerson Harkin is from the Nod Lab, just getting ready to present.
Hi, can you see me?
Yes, I can. Hello.
Hi. How do I share my screen?
So you go to the top of your window here, and perfect. Here it is.
There we go. Great.
So I will bring it into focus, and we're ready.
Okay, fantastic. Well, thanks for everybody for turning out to my talk. My name is Emerson Harkin. I'm from Richard No's lab at the University of Ottawa, but this work has a lot of experimental work that was carried out in collaboration with Jean-Claude Biek's lab, also at the University of Ottawa. So I suppose I should thank the previous presenter actually for giving such a nice introduction to the serotonin system. And I'm going to go kind of very briefly over this again, as I'm going to be talking about a couple of kind of network mechanisms for gain modulation in the serotonin system. So in particular, I'm going to be talking about the dorsal raphe nucleus, which is a really small midbrain nucleus that provides most of the serotonergic input to the forebrain, and plays a big role in regulating a lot of different behaviors. I tend to mostly be studying mice, so the behaviors that I'm most kind of concerned with are eating cheese and running away from things if you want, but obviously it's involved in regulating a lot of different things. So the serotonin system, or at least the dorsal raphe nucleus, is mostly composed of serotonin neurons, but there's also a pretty large population of somatostatin expressing GABA neurons, which send these feed-forward inhibitory inputs onto 5-HT cells. Now, I mean, in spite of feed-forward kind of inhibition being kind of a widely studied network mechanism or network architecture, in the case of the dorsal raphe nucleus in particular, the kind of cellular properties, distinctive properties of these two kind of cell types mean that we don't really have a great kind of sense in the field of how the interactions between GABA and 5-HT cells are necessarily shaping the output of the serotonin system. So the goal of this project was really to try to shed a little bit of light on that process. So we basically started by making patch clamp recordings from 5-HT, genetically identified 5-HT and GABA neurons in dorsal raphe nucleus of mice. We found that 5-HT cells have these kind of characteristic slow firing patterns that are heavily influenced by this really large transient potassium current, and that the firing patterns of GABA neurons and their intrinsic currents were a little bit more diverse. So to really understand how these kind of cellular properties influence the network output of the raphe, we started by basically building a set of really strongly experimentally constrained single neuron models. So the modeling framework that we used is based on generalized integrated fire models, which it's kind of easy to extend with basically explicit models of particular, biophysical phenomena of interest. So in our case, we were really interested in this potassium current in serotonin neurons. So we built a model that can accommodate that, and basically that because it's built on top of this generalized integrated FIRE framework, can be constrained to a global optimum. So without going into too much detail into kind of the fitting, model validation procedure, the main thing I want to point out here is that these traces are sample traces from, I guess, real neurons and some corresponding model predictions. And if it's not immediately obvious which one is which, then hopefully that means we did a good job in our modeling. So to kind of understand, at this point, how the dorsal raphe kind of network behavior emerges from the properties of single neurons, we basically took our banks of models that we had fitted to individual raphe cells and sampled them with replacement in order to create models of large dorsal raphe populations. So armed with these population models, we applied some kind of step inputs basically to look at the sensitivity of these populations to inputs over time. And we quantified that sensitivity in terms of the gain. So in particular, basically, the gain is the slope of this population FI curve. We give inputs at different amplitudes, and you quantify the slope of the population firing rate. So we found that in our simulated dorsal raphe nucleus network, that serotonin neurons responded basically very strongly. They're very sensitive to kind of sudden, fast inputs, and that the overall amplitude of the serotonergic output is basically regulated by the amount of feed-forward inhibition. So if you either remove GABA neurons or if you kind of decrease the strength of their inputs, then you can kind of overall change the sensitivity of the dorsal raphe nucleus to its inputs. Now, interestingly, if you change the amount of the current that the potassium current that we found in serotonin neurons earlier, you actually selectively can change the sensitivity of serotonin neurons to kind of fast, sudden inputs without kind of affecting its responses to more prolonged inputs. So we basically found these, or in our simulated populations at least, these two kind of separable mechanisms for control of the dorsal raphe nucleus over a couple of different timescales. So with that, I want to thank everyone who kind of participated in the project, and to you, everyone in the audience, for listening in. Any questions?
Great, thank you so much. So I invite anyone to write down questions in the panel below. So I have one question. So very interesting results. Given what you found, what would you predict would be kind of the key interactions between the serotonergic and noradrenergic systems with respect to stimulus processing?
Right, that's a really great question. I mean, unfortunately, I have to be a little bit kind of speculative because we haven't really. It would be nice to repeat this kind of work in the noradrenergic system, but for now, what we can kind of speculate on is that it really seems to be the case that this kind of sensitivity of serotonin neurons to kind of sudden, fast inputs is really under control of this transient potassium current. And there's some evidence that the amplitude of this transient potassium current is regulated by noradrenergic input. So what we think is really kind of potentially the key interaction is that basically depending on, in what kind of behavioral or environmental contexts, it's really critical for serotonin to respond kind of acutely to things, maybe during learning or when something kind of surprising happens in the environment. Then it would be kind of the role of noradrenergic input, maybe from the locus coeruleus, to kind of tune the extent to which the serotonin system is really going to respond kind of strongly to those sudden events. we think maybe.
Okay, we have another question related to model optimization. Dylan, do you want to ask your question?
Yeah, of course. First, thanks, Emerson. Cool talk. And I'm always curious about how people fit their data to models. I'm thinking about how to do that for myself for different type of data, but still, I'm still curious about how you do it with your generalized leaky fire and integrate model.
Right. Well, thanks for the question. I'll just switch to one of my backup slides here. So the definition of the model is kind of on screen here for anyone who's kind of a technical aficionado, I guess. But the main kind of important thing basically is that the model is kind of broken into two parts, and one of the parts is this subthreshold rule, which is linear with respect to the parameters. So in particular, we provide basically this input current, and we have from our recordings basically the voltage of a neuron over time. Mm-hmm. If you have the voltage over time, then of course you can calculate the voltage derivative really easily numerically, and everything else is just a scaled version of that basically. So this is a really easy thing to fit just with like multiple linear regression.
Right, least squares.
Yeah, exactly.
Right.
And the second part, we have to fit by maximum likelihood, and there's not really like a closed form way to do it, but there are some nice proofs that the likelihood function at least is convex, so it's easy to do numerically.
Right, so some sort of gradient descent and to maximize likelihood.
Yeah, exactly.
Okay, right on. Cool. All right.
Thanks for the question.
Okay.
It's all good.
Thank you so much. Thank you very much for the presentation. We will go to the next presenter, Marcin. So Marcin, I will invite you up to the screen. Okay.
Hey.
Hey, so to share your screen, just go to the top of the window and click on the second last icon from the right.
Well, it's a share the event on social media. I don't think I can see that.
Are you on Chrome?
I'm on
Google Chrome?
Yes, I'm on Google Chrome, yeah. Oh,
you
okay.
Are on...
Okay, wait a second. I think I see it now. Okay.
Okay, perfect.
Can
yes.
you see my screen now?
Yeah, we can see it.
Okay.
Take it away.
Okay. So, thank you for organizing this meeting, and thank you for letting me to present. Although I'm not really affiliated to any Canadian institution. So my name is Marcin, and I work with Charlie Schroeder at Columbia University and Nathan Klein Institute. And I'll show you some of our results on the physiology of broadband high frequency activity, also known as high gamma. So the broadband high frequency, or BHA, is this broadband shift in power spectrum. So for example, if you place an electrode in the monkey or human primary visual cortex or auditory cortex and present an animal with different stimuli, what you might see, and you've heard that a lot during today's session, you might see this narrowband gamma. But on top of that, you will see this broadband shift in power spectrum. Well, this BHA or high gamma, I prefer to use the BHA not to make any associations with the narrowband gamma. So this BHA signal is a very prominent feature in intracranial recordings, particularly, very useful in human electrophysiology. It's not only observed across species, but also across cortical areas, so like V1, A1, and also observed in the medial temporal lobe. It's also functionally relevant in a sense of it's being modulated by several cognitive functions. And it's also probably the best correlate of multi-unit activity. So it's not only offers us translations across animal models, but also across recording modalities. What is particularly interesting, I guess, for some people at this meeting is that it's also useful in terms of decoding. So it's been shown that it's very information rich. And it's also impaired in schizophrenia and other disorders. But at the same time, we have very little understanding of the physiology of this signal. So there is beautiful work from Supratim Ray and colleagues who show that this BHA here presented in red is nearly perfectly correlated with multi-unit activity. And importantly, when you look at other frequencies, this correlation breaks. So this signal has been frequently taken as a proxy for multi-unit activity. And so we decided to have a little bit closer look at the laminar generation of this signal. So we inserted this multi-layer, multi-electrode linear probes into primary visual and primary auditory cortices in macaque monkeys. So we could record 23 equally spaced contacts, 23 field potentials, from which we could calculate the second spatial derivative, which gives us the distribution of sinks and sources. And what we know from previous studies that especially when we combine it with multi-unit activity, for example, the current sink over the source is where the layer 4 is. So this gives us a pretty good physiological understanding of this canonical microcircuit, and we could use that to try to understand how this BHA signal is generated. So what you see here is on x-axis is time, on y-axis is the cortical depth from the PL surface to white matter. The 0 time point, in this case, this is recording from V1, is a brief presentation of the whole screen flash. And what we observed, actually to our surprise, is that the BHA has these two components, so early deep and late superficial. And this was surprising because we know from previous studies in monkeys, but also other animal models, that the multi-unit activity is actually, or firing is actually strongest in the deeper layers. Firing in the superficial layers is fairly sparse, especially with the type of stimuli that we used. So when we compare the response from the baseline for both multi-unit activity and BHA, it was surprising to us that we saw this pretty strong and fairly well overlap in time and space between the early deep BHA, but not so much between the late and superficial. And this is important because building on this assumption that the BHA is a nearly perfect homologue of multi-unit activity, we're expecting totally opposite pattern of results. And this is just one penetration. So one time, one experimental session where the electrode was lowered to the cortex. This is the example I just walked you through. There is more examples like that, and I just like to show you a very simple few examples so that you could get a taste of how clear the dissociation is. So that's another penetration where you see again the multi-unit activity being strongest, but around layer 4 and deep layers, and BHA having again these two components, so early deep and late superficial, and only early deep is in fairly well correlated of multi-unit activity. And that's another penetration, and I could keep on going for a few minutes. So I guess I'll just skip to the summary. So what we observed is that there is these two components of the BHA. So it doesn't really follow the laminar distribution of the multi-unit activity that we know. We tested also several other questions that I'd be happy to move offline if you're interested. But one interesting question was so which of these two BHA components volume conduct to the PL surface? So when you do human-like ECOG recordings, for example, with the surface electrodes, which of those two signals volume conduct and which of those two you pick up? And actually, from our analysis, it seems that both of them volume conduct, but the superficial signal, of course, volume conducts to a larger extent. So the ECOG-like signal is dominated actually by the superficial layers signal, which is not that good of a proxy for multi-unit activity. We also did some pharmacological study where we looked at the effect of low doses PCP, so non-competitive NMDA blocker, and we found that hints of pharmacological dissociation. So at this low doses, the BHA was already attenuated, but the multi-unit activity was intact. And then we test whether this dissociation generalized. So we use different stimuli in both V1 and A1. So in V1, we use passive and active vision. So passive vision is just passive stimulation, passive presentation, or presentation of arbitrary stimuli. While active vision is fixation locked potentials during free exploration of colored images. And in A1, we looked at the best tone and broadband noise. And we also had this subset of laminar recordings in human that show similar effects. So of course, this is a very collaborative project across that was conducted at both Columbia and Nathan Klein, together with NYU, Bob Knight from Berkeley, and we also had, as I mentioned, a small sample of data with a similar laminar recording in human that was pioneered by Istvan Ulbert. And well, the paper is actually available in bioRxiv if you're interested. And thank you.
Thank you so much. I will leave your slides up in case there's questions related to specific slides. So I have one question. So these two components that you talked about, how does their expression relate to NMDA receptor plasticity and antagonism? So you showed some drug-related effects. Can you elaborate on that?
Yeah, so in previous rodent studies, the prominent superficial sink has been observed to be NMDA dependent. And that time and space where the sink is strongest actually corresponds to our strongest BHA component. So we thought that it might be, we were just kind of blindly trying to connect those two findings. And then we happen to have again, this is a very small data set with one animal, a few penetrations only. And with this non-competitive PCP antagonist, NMDA antagonist PCP, and so that's the connection, I would say, right? So there is also other sources of evidence pointing towards preferential the density of NMDA receptors is highest in the superficial layers. So that's another source of evidence that pointed us towards linking those two phenomena. But again, this is very preliminary. So it's just 7 penetrations, and we take it rather as kind of anecdotal at this point.
Okay, thank you very much. We have another question. Does predictive coding help in understanding early deep followed by late superficial BHA activity?
Yeah, that's a very good question also. So this is a canonical microcircuit that we've been using that has been already mentioned in predictive coding several times today, I guess. And there's two interesting things, I would say. So this high gamma or BHA has been suggested to reflect primarily the feed forward, whereas our data suggests it's strongest in the superficial layers. So this is where feedback projections terminate. So it suggests that it might be actually some portion of this signal, particularly the superficial, might be weighted towards feedback projections. The temporal difference itself is probably just reflects the difference across cortical depth. So the early deep BHA corresponds to the layer 4 and infragranular layers, which is the thalamic input. So that's where actually signals from the LGN hits the cortical column first. And then the interesting question, and we don't really have answers, whether the late superficial BHA reflects monosynaptic drive of the superficial layers, or whether this is a top-down feedback from higher order cortices. So that's at this point, I would say this is equally possible, and that would, probably the later is actually more likely given the fairly long temporal difference or distance between the early deep and late superficial.
Okay, great. Thank you so much. So actually the person asking the question is the next presenter. So I will invite him now on screen. And thank you so much.
Okay. Well, thanks, Martin, for that answer, actually. So good timing.
So to share your slides.
Yeah, sorry, I clicked on share, and I have this document, but that's not showing up.
So share screen. If you click on share screen, you should have a window that is either your entire screen or just the application window, and then you can choose the application that you want to share.
Okay, so let me try this.
Okay, looks
okay,
good.
So you see this?
Yes.
Okay, excellent. Okay.
And
so,
you can click, you can click hide, exactly.
Right,
Perfect.
okay, thank you. Well, thank you, Andrea, for taking time out of your day to host this set of parallel talks. It's very appreciated, and it gives us a chance to talk about our work. And today, what I'll be showing is less about it's less about empirical work and more about how I think about simulating amplitude dynamics as seen in electrophysiology. So more of this presentation will be building up the intuition I use, the mathematical reasoning I use to come up with a different model of amplitude dynamics. And I thought that perhaps a first starting point is to make contact with the concepts of forces and energy potentials that are pretty foundational to classical physics. So loosely speaking, the idea that a forces drives a system as though it's performing a negative gradient descent on an energy potential is pretty useful to me. And hopefully this will provide insight to you as well. But to make this real, let us look at a specific example. And there's a reason for this specific example. And I want you to focus on this alpha parameter. And this alpha parameter basically alters the general shape of the energy potential. So let's look what this actually looks like. So pretty simple. If we have negative values for alpha, we have this boring single well potential. And what this says is that wherever you see a local minimum by the virtue of this relationship is where your solutions are going to converge towards. And if it's convex, it means it's not only going to converge towards this point, but it'll be stable around this point as well. But if you turn this alpha value to positive values, you'll have a double well. So it means you have 2 points where this is the case, where you have these stable fixed point solutions. And this is funny because if you remember Scott's intro slides, he talked about this bistability regime. So it's funny that I was pretty fortunate to have him introduce some of these concepts. But if we spice it up a bit and allow these potentials to rotate along this vertical axis, we get an interesting set of behaviors. And this is because the difference between the single well, spinning single well, and the spinning double well is underlined by what is called a supercritical Hopf bifurcation. And it's not meant to go deep into supercritical Hopf bifurcation, but more for this figure that lets us understand what's going on. So what we see here is as though we were looking at these spinning potentials and the evolution of these solutions from a bird's eye view. So if you were looking from top down, okay, from a bird's eye view, and you see that for negative values of alpha, you just get this boring convergence to a steady point. But for positive values of alpha, you get this convergence toward what we call a limit cycle. So this is just a simple way to actually model oscillations, because the trajectories will just go around and around this limit cycle indefinitely. And if you use this specific set of equations in polar coordinates to simulate this transition between a fixed point and a limit cycle, you're dealing with what's called a Stewart-Landau oscillator. There's many terms for this type of oscillator, but this is the one I like, and you'll hear me use it quite a bit. And to make things a bit more interesting, we could drive the dynamics of the Stewart-Landau oscillators using some noise. So you let the solutions sort of explore the energy potential landscape. And you see here, for instance, in the double well, some sort of switching behavior between both solutions. But what I really want to talk about, so this was a bit to build the intuition, and what I really want to talk about is if we take back the same energy potential, but then let this alpha parameter be something that varies as a function of time. And we look at a concrete example. So say we have initially a positive alpha value, so we start with a double well potential, and then we have this potential transition into a single well potential by having alpha be negative, then it stays there, switches back to double well, back to single well, and then whatever, so on and so forth. Well, this time varying behavior of the shape of the potential, in this case, was just modulated by some arbitrary signal. If this was just done using PowerPoint. But in reality, we could use any signal, including a Stewart-Landau oscillator itself. Okay, so to me, I can't help but to analogize this to the movie Inception, okay? And to be sure that this analogy makes sense, let's really look at an example. What I've been working on a bit is we have these two curves, okay? And they're two oscillators, really, one in orange and one in blue. And they're both driven by noise-driven Stewart-Landau oscillators, okay? What we see in blue is a fast subpopulation that we'll call, that have very high, like a higher frequency. And in orange, we have a slow subpopulation. And what we notice is that every time the dynamics of the the slow subpopulation peaks, the amplitude of the dynamics from the fast one will peak as well. And this is not surprising because alpha of the fast subpopulation, which alters the shape of the potential, is directly dictated by the slow subpopulation, okay? So this is really all I wanted to show. It's pretty simple, but it's just good to know for anybody that just wants to model amplitude dynamics using a quick set of simple rules. And I thought I'd just finish with this beautiful scrapbook of signals to just show you that you could recreate actually pretty realistic data. Some more like sustained oscillations like here, some more maybe like less sinusoidal oscillations could be something interesting as well. So I just thought that, oh yeah, that was another idea, but I thought I'd drop that. So that's it.
Okay, thank you very much. Let's see if there are any questions. In the meantime, I will ask one. So the oscillator that you presented, could it be a useful, so if you could comment on whether it could be a useful way of modeling bistable percepts? Oh. Did the connection freeze? Okay, so I think the connection froze and maybe we lost Dylan for the moment. Just waiting for him to come back here. Now, just for the audience, does anyone have any additional questions or comments before we end the session? So there are still some questions from one of the previous talks. If you'd like to still address it, could you vote it up? Emerson, would you be interested in answering the question to your talk? I can invite you on the screen.
Sure. I actually thought that was for a different talk.
Perfect.
Yeah. So the question is, would autoreceptors act as a slow or a fast feedback? I guess in the dorsal raphe nucleus. So autoreceptors are kind of an interesting. I guess.
Yeah. So yeah, sorry about that. I think my connection completely dropped out for some really weird reason. So
no worries. So we just wanted Emerson to answer one of the questions that was posted, and then we go back to you, if that's okay.
Yeah, no worries. Yeah, I'm sorry about that. That's my.
No worries.
I don't know. I really don't know what happened. That was so weird. But anyways.
I don't mean to steal your thunder there, Dylan. So
no, that's fine. Look, I mean, and there's like we have some time left, so take your time. It's fine.
Right. So the question is, would autoreceptors act as a slow or fast feedback? So serotonin neurons in the dorsal raphe nucleus express some inhibitory serotonin receptors themselves. The kinetics of those receptors seem to be a little bit slow. So I think they activate on the order of like hundreds of milliseconds and inactivate over seconds. So from that perspective, it would be kind of slow. But some experimental work in Jean-Claude Beek's lab, which I think there've been a couple of posters at SFN about this at different points. I'm not sure if there's a bioarchive yet. But it seemed to show that these actually act mostly as heteroreceptors. So it's not clear that they're acting as kind of a simple form of negative feedback. But if they were, it would probably be slow. Yeah, thanks.
Great, thank you so much. So just before we had the technical difficulty, I was asking the question for Dylan. Your Stewart-Landau oscillator, what type of behavior do you think one could model using this framework?
So yeah, I don't know where it cut off. So the idea here is that what I was showing is a nested structure where you have one subpopulation of oscillators affecting the activity of the other. So if we break this down in the following sense, that if we just take one population, okay, the most basic case, what you could do with that is when that alpha parameter is 0, the bottom of that wheel becomes very flat. And when that becomes the case, and you have a noise-driven system, there's like a bit of an itinerary behavior. And what you get, in a sense, is these sort of transient bursts of oscillations. Okay, the reason because you have that potential and it's flat, but it's still bounded. You still have that convexity to keep it within your steady state. But that steady state is not really one where your solutions will tend to like really converge very strongly towards. So you could get like these very intermittent behavior where like you have some noise, basically some sort of noisy dynamics, and then for a few seconds you get a clean oscillation, and then it subsudes back. And the idea here was to just make sure that that specific behavior, that balance between oscillatory behavior and noisy behavior, is controlled explicitly using another population of Stewart-Landau oscillators. So you could have like these transient bits, transient rises of the sort of excitability of that other subpopulation. So you'd have times where the dynamics are like strictly noise dynamics, sometimes where it's a bit of both, and then sometimes where it's purely oscillatory, and then you can modulate that directly. Anyways.
Great, thank you so much. So we have a question. What does the energy landscape of a damped oscillator look like?
Of a damped, so the thing is that we have it's a potential, but it's spinning. Okay, so the only thing that's accounting for the oscillatory behavior really is the fact that the potential is spinning around an orthogonal axis. So what could be, so a damped oscillator, for instance, if it would just be this, actually, okay, so might as well. Can we see that? No, I'm not sharing right now. But basically that single well potential I was showing. So maybe might as well just do this real quick. Okay, do people see this slide?
Yeah.
Okay, so there's just this single well one, okay? The single well potential. If it's spinning and you have a solution that starts up here, it's going to converge to the bottom of this well. But the fact that it's spinning still sort of promotes, it translates into oscillations. But the fact that after some time is just at a zero altitude, if you will, zero energy, it'll stop oscillating, even if the potential itself is oscillating. So to answer the question explicitly, it would be this sort of energy potential, but you would initialize the solutions to be higher up the well. So you could even perturb your solutions to go up the well and let it relax down. Perturb, up, down. You could do that as well. So it would really be the single one, the single well.
Okay,
So,
thank you. Thank you so much. If there are no other questions, we will close this session, and I'll invite everyone to join me for the concluding remarks. Thank you so much to everyone for the wonderful talks.
okay, thank you, Andrea.
Thank you.